So I will follow a little bit in the footsteps of Badri and other talks today. In fact, that I'm going to talk about histopathology and actually cytopathology. And unfortunately, the method which I'm going to talk about has a much shorter history, so I can really summarize everything over the last 5 or 6 years. That's when things really started to pick up there in this field. And it is the methodology we're going to talk about, and I'm going to do a little bit of a historical perspective here, is, as I mentioned before, in cytology and histology. In cytology, the problems are even harder than they are in histopathology, because you're looking at individual cells. The cells are exfoliated. Typical tests are, for example, the cervical cancer screening, which is done about 100 million times a year in this country alone. And here a cytologist looks at a deposit of about 10,000 cells on a slide. And the pathologist looks at 3 or 4 cells which are abnormal. So just the chance of finding 3 or 10 out of 10,000 is pretty slim. And what they're actually looking for is not that ugly clump here, which is actually just lymphocytes. It is actually this particular cell down here, which you can see, which has a larger nucleus. So the cytopathologist looks at differences in nuclear morphology, cell morphology, different nuclear to cytoplasma ratio, and also in changes like the nuclear membrane. And you see here that nucleus, it has, well, looks a little bit coarse in the membrane, and this is actually what is looked for. In histopathology, as you have heard before, problems are a little bit easier because you have not only the cellular morphology to work with, but also the architecture of the tissue. It was not supposed to happen. In both of these methods, the diagnosis is subjective. It cannot be easily reproduced. If you give the same sample to 2 cytopathologists, you get very rarely the same result. And therefore, optical methods have been developed, and you have heard a number of them, to increase the reproducibility and be objective, operator independent, and so forth. You have heard about fluorescence. That is one way to do it. You can do it through light scattering. That has been done quite a bit with standard microscopy. But what we are trying to do is use methods of vibrational spectroscopy, infrared and Raman spectroscopy. Why? Because in fluorescence you get one broad, ugly peak in the spectrum. In vibrational spectroscopy you get hundreds of peaks. And they contain a fingerprint of the chemical composition, and this is what we're trying to exploit here in this work. I started this work in the mid-90s when I was at the City University of New York. I came here in 2006 and started the laboratory for spectral diagnostics. And the idea is, here we want to collect a complete infrared spectrum of a cell or from a pixel of tissue. The infrared spectrum is a snapshot of the total biochemical composition, and therefore contains really genomic, proteomic, and metabolomic information. We can see in the spectrum, for example, whether glycogen is metabolized by a cell or not. That is very easily detected. And we collect thousands and thousands of spectra and analyze them by methods of multivariate statistics. And what we're shooting for example, in a population of cells, that the cells which are coming from normal and from abnormal donors separate quite nicely based on the chemical composition, and we know nothing else about the cell. This is done completely unsupervised. This is a typical result which we get. And similarly for tissue, this is a 1 by 1 millimeter tissue section on the left, the stained tissue, the way the pathologist would look at it. On the right, the spectral image obtained from infrared spectroscopy and again from unsupervised multivariate analysis. That means we know nothing about the tissue, just we compare tissue spectral properties, and we segregate the tissue areas into the different areas which are similar by chemistry. So we have here a new method which can be used for diagnostics. It can be used as an adjunct method. It can be used obviously as a diagnostic method. So let me just very briefly tell you where we were in 2006 before I came here. And that was actually our first very, very exciting result. We are having here samples from canine cells and human cells. No cytologist in the world could visually distinguish them. They look absolutely identical. I mean, a dog, after all, is a mammal and is a relatively close relative of ours, okay? And we could distinguish them. We have about 1,000 individual cells. Each symbol represents one cell, and there's one misclassification in this. This is orders of magnitude better than what a cytologist could do. And this, in this particular case, was based on the metabolomics. We can see that the canine cell metabolized glycogen differently than the human cell. And this was one of our first successes in detecting unsupervised, by unsupervised methods, cancer in the lymph node. This is an excised lymph node, about 10 millimeter in diameter. The area in the green circle is actually the cancer, and our spectral diagnostics very, very nicely, unsupervised, without input from any pathologist, found the tumor. So that was in 2005, 2006. The state of the art then was painfully slow. For example, in the cytology, we had to select individual cells, localize them in the microscope, put an aperture around them, orient the aperture, store all this information. We could do about 200 cells a day. In the tissue, we could perhaps collect 10,000 spectra, but it took us a day to analyze an image. So it was a very slow process, and it was obviously good as a research tool, but it was not ready for commercialization. Then, for some reasons, I got an offer here from the Department of Chemistry and Chemical Biology at Northeastern. Still don't know why, but perhaps somebody saw the potential for real-life research. Perhaps somebody saw the potential for intellectual property for the university. And there was, of course, a very close potential interaction with CenSSIS in terms of the image analysis. After all, it doesn't matter whether you look at a Landsat picture and look at a pixel 10 by 10 meter and analyze it for the composition, or you look at a piece of tissue which is 10 microns by 10 microns. Same problem. Probably didn't hurt that I had 2 NIH grants at the point. And since moving here, it has been just a very, very successful time in terms of 3 patents, book chapters, and a number of papers. Then the next thing, of course, was how do you get something like this to market? And you will hear later on Stan talk about his view of this. And I think I can summarize this as automation, standardization, and acceleration. There's no sense to do a test where it takes me a week to calculate a diagnosis, okay? So the first thing was to, instead of sitting in front of the microscope and select cells visually, we automate this completely. We're scanning right now a large area of a tissue deposit or a cellular deposit. It gives enormous data sets. We patented this approach in 2008, and this entire method is now automated. Cells are stained, imaged, stored with the spectrum in a database, and we have collected about 500,000 spectra so far. And the interesting news is here that this method distinguishes cells which are absolutely visually indistinguishable, but come from patients who are normal or abnormal by cytology. and we distinguish them as blue and red. And even we can distinguish whether a person had a history of cervical disease. And that was something very unusual. First of all, we see the spectrum of abnormality in all cells, whereas the cytologist only sees it in 1% of the cell. And we have this history. And it was actually our pathologist at Tufts Medical Center who pointed out that this is due to the viral infection, which is the